All right, everybody, we're going to go ahead and get started with our Thursday night guest lecture. Ashley, can you mute this so we don't hear ourselves? Or is it this?
We should go to volume down using that.
All right, how's that sound for everybody in the room? Perfect. All right. All right, we're going to go ahead and get started in a few minutes. Just some notes. Thank you all for your patience this week. It's been challenging with the weather and rearranging schedules, so thank you all for that, students and professors. Remember, checkout for the 1 week classes is Sunday morning. We will start running boats off the island starting at 10 o'clock. 1 week students have to be off the island by 1 p.m. And my students, I would like to meet with all you for about 5 or 10 minutes after this lecture downstairs in our classroom to go over just a few things. Does anybody else have any updates? Tori or the other professors? Nope. Okay. All right, so we're going to go ahead and I'm going to ask Zach to introduce our speaker.
All right, it is my pleasure to introduce Dr. Ieva Roznere. She comes from Binghamton University, where she did her undergraduate work. And just like the RU students we have here this summer, she actually did an RU, but at a different university. So I would implore everyone to look into the RU programs at different locations. She did her RU project at the Cary Institute of Ecosystem Studies. And her project was on the distribution and historical record of chlorophyll A. In a lake in New Hampshire. She then went to complete her PhD at The Ohio State University with Tom Waters, where she studied the health assessment of freshwater mussels using metabolomics. And she currently has an appointment in EOB, which is a department at Ohio State in the College of Arts and Sciences. And she has an appointment with the Columbus Zoo as the science director of the Columbus Zoo and Aquarium Waters Aquatic Conservation Center. So I will hand it over to Ieva, but we will get started.
Thank you so much. I want to thank Stone Lab first for inviting me, especially Zach Sutton-Snyder and Justin Chapin. I was here a few years ago, several years ago, so it's nice to come back and visit and see the place again. So thank you for that opportunity. And today, I'm going to talk about freshwater mussels. I know many of you probably know quite a bit about freshwater mussels, but I'm going to assume zero knowledge because a lot of people don't know about them. So I'm going to teach you guys about their biology, what they are, why they're endangered, and some of the conservation work that we're doing. Yeah, thank you. Okay, so I split this up into 3 parts. The first part is where I ooh and ah you about the amazing facts about freshwater mussels because they are very interesting. The second part, I'll talk about the different. The last part will be about the different projects, the research that we do at our mussel facility. So, freshwater mussels belong to the group mollusks, right? So that encompasses pretty much bivalves, the snails, even the octopus. In that, they are bivalves, right? So anything with 2 shells is called a bivalve. And within that group, they are the Unionidae family. So the term freshwater mussels is actually a very specific term that applies to this family. You know, zebra mussels live in freshwater, Probicula live in freshwater, but they're not called freshwater mussels. So I just wanted to kind of clear up if there's any confusion about that. I know it's kind of odd, but the term freshwater mussels has become so common, even in the scientific literature, that that's just what we call them. But they are a very unique group of animals, and they are all found in our rivers and our lakes. So they're not even the marine ones that you eat in restaurants, and they're not zebra mussels, right? So those are invasive. But the native mussels that are found here are called the freshwater mussels. And we have about 300 species in North America. A lot of people might not have heard of them because they're mostly buried, they're hard to find, hard to see, and there's not a lot of behavior for people to notice. So they kind of seem a little boring. But they are actually the most endangered group of animals in North America. And that's according to Fish and Wildlife Service. And a lot of people are surprised because when they hear endangered, they can think of amphibians or birds or things like that. But if you look at the percentage of the species in this group that are threatened at some level, these are the most endangered group of animals. And I put up some pictures on the right just to showcase the diversity of shapes and sizes. All the ones there, except for the one with the spines growing out of it, these are all native to Ohio. So they also have fun names: Three Ridge, Rabbit's Foot, Pistol Grip. They have fun names. But I put that one up there, even though it's endemic to Georgia. It's only found in Georgia because it's really cool looking, and it just shows how different they can look. Okay, so here is an image of a mussel that is buried in the sediment or the sand and rock in the bottom of a river. So there are some examples just to kind of show you guys what they look like. But they spend most of their lives mostly buried. They have a little bit of their body sticking up because they have to filter feed, right? So the little gaping holes you see, those are the inhalant and exhalant siphons. They do have a foot. It might not be what you consider to look like a foot, but it is this little blob of muscle that comes out that does help the mussel move. So it can actually move. It doesn't do large distances, so it's considered fairly immobile, but it does have a foot, and that's what it uses to bury itself or to move a little bit. And then they are also fairly long-lived animals, and it's easy to kind of clump them together into this one group, freshwater mussels, and think, oh, they're just this one thing. But really, they are very different according to different species. So some species only live 5 years. Some in Eurasia live over 100. So for Ohio, the average lifespan is between 15 and 40 years, but it really depends on which mussel we're talking about. Then this is probably the most interesting thing about mussels is that they have a very unique life cycle. So this life cycle that I'm going to describe is unique to freshwater mussels. It's not found in any other bivalve. So no zebra mussels, no marine mussels, no plants. No one does this, but freshwater mussels have evolved a life cycle that includes a parasitic stage on the fish. So we have male mussels and female mussels. The males will release sperm into the water column and these little sperm balls that kind of float around. As the female is siphoning the water, she'll uptake some of those. And when the eggs are fertilized, they develop into larvae. And we call these larvae glochidia. There's a little zoomed in version, and it's basically superficially a little mussel. The shell actually never changes when they transform. But these larvae are brooded in the mussel gills. So the gills are a huge organ, and a part of the female gills are used to brood the larvae. And when they're ready, they have to attach to the outside of a fish in order to transform into juveniles. So they'll attach the gills usually, sometimes the scales or the fins. But that's where they sort of insist into a little ball and transform. They don't grow much. Mostly their internal organs transform. And when they're ready, they drop off and begin their life as an adult. So this does not hurt the fish much at all. It's called a parasite because obviously the fish does not benefit, and the mussel does take a little bit of nutrients from them, but it's not a disease. It's not a parasite like you can think of causing disease. It's thought to have evolved more as a means of dispersal, right? It's a way for the mom to get her babies elsewhere, because otherwise all the mussels would just kind of eventually end up downstream. And so this is a way that they can disperse. And not any fish will do. So some mussels are considered generalists. They can transform on a wide variety of species. And some mussels are called specifists, and they can only transform on one or a few species. Some only transform on carp or catfish. And then there's a huge range in between, but there's always some sort of specificity, and it's unique to each species of mussel. So if you imagine the chance of those little larvae, the Glochidia, attaching to a fish and they're just floating around the river, it's not very high. So the mussels have evolved ways to attract the fish to come to them. So the same way as fishermen use lures to attract fish by fooling them into thinking they're getting a meal, freshwater mussels do the same thing. They have different lures to attract fish to come. And they trick them. And so here's an example of a mussel that's buried. That was sticking up. Oh, and I forgot it was gonna, oh. I was going to actually do it here because it's better quality. So this is an example. Right. I knew this might be an issue.
Oh, you're good.
I have the videos embedded in the PowerPoint, but they were kind of slow. So we decided to try YouTube. So this here is a mussel that's slightly sticking up above the sediment. And the little flaps that you see there are the mantle. So the mantle is sort of the covering that lines all the organs, and it's next to the shell, and it sticks up and it resembles a fish, right? So you even have the little eye spot right there. You have a little tail, and it looks like a little fish swimming in the river. And you can see how similar it looks to the actual fish surrounding it. And in between the little flaps there, you see those are the gills. So when the gills are really plump, they're filled with the Glochidia, and the fish will bite down on the mussel and think it's getting a meal, but it's not. It's getting all of those Glochidia attached to it. So this here is the rainbow mussel. It's actually quite common in Ohio, and this one is pretending to look like a crayfish. So you can see the eye spot and the legs, and it will actually move itself in a way that makes it look like the crayfish is crawling on the bottom of the river. And so obviously this one's targeting fish that will attack and eat a crayfish. And again, the larvae don't taste good, so the fish will spit them out, and it's that flushing back and forth on the gills that lets the babies attach to the gills, because they don't want the fish to eat the babies, right? They're going to die if they do. They're external parasites. And so the last one is the riffle shell. This one is a federally endangered species. We have it in Ohio, but it is very rare. So this one doesn't actually mimic anything. It is just gaping, and it's very white, and it looks very interesting. And so a small little fish, a darter in this case, might get a little curious. It might poke its head inside to see what's there, and the mussel will clamp down shut on the fish. It then holds the fish and it pumps water through its gills, and that's how all the larvae attach to that fish. Again, it doesn't harm the fish. It does this for 10 minutes, and then it lets the fish go. So this is, like I said, a federally endangered species. The only reproducing population we know of is in Pennsylvania in the Allegheny River, but we did move thousands of these into Big Darby Creek in Ohio. So be careful when you walk with your toes. The only thing that can happen in this case is that if the mussel closes on the fish in such a way that the gills are closed, the fish can suffocate. That can happen. It's obviously not ideal, because then your reproductive attempt is a failure. But in this case, it opens up and the fish is a little bit dazed, but it eventually swims away. Every mussel in this genus is federally endangered, so it's probably not the best approach, But there's a lot of different kinds of lures. Some mussels will package up their larvae into little things that look like worms and spit them out, or some of them are attached. Some just spit out these mucus webs that float around. So there's all kinds of different lures used to attract fish. Okay. All right, so this one is one that did not survive in our lab, but using that same northern ripple shell. Unfortunately, the mussel had closed on the fish's gills. But all those little white dots that you see surrounding the four little eyes and the mouth, those are all the Glochidia. So they're technically not microscopic because you can't see them, but they look like little grains of sand. Okay, so that is the biology of mussels. Next, I'm going to talk about their threats. The first time that humans negatively impacted mussel populations was back in the 1857. This is when the period called the Pearl Rush began. So what happened was there was a carpenter in New Jersey who was just sitting on a log in a river, just shucking mussels for no reason that I know of, just bored, I suppose. And he ended up finding a 1/2-inch pearl. Now, I should mention that pearls are not unique to freshwater mussels. Pearls can be found in any bivalve. And what a pearl is, it's actually an immune response. So if you imagine the mussel is slightly gaping because it's filtering water, if there's a little irritant that gets inside or a little parasite and it kind of gets stuck between the mantle and the shell, there's no way for that mussel to get rid of it. You know, it can't just kind of wipe it away. So the same material that covers the inside of the shell, that mother of pearl material called nacre. It's the same material that's constantly being produced as the shell grows. They will overlay that little irritant with a thin layer of that nacre and then keep doing it forever. And so after years and years, you get a pearl. So the irritant is actually in the inside of that little pearl. So you could find a little parasite or something. So if you've ever seen real pearls, they don't look very pretty. They are asymmetrical, different colors because they're genuine. Okay. So this guy found a 1/2-inch pearl and he sold it to Tiffany, the same company that we know of today. He sold it for $1,500, which I believe in today's money, it's 17,000, something like that. And the word got out in the newspapers. It became known as the Queen Pearl. And the California gold rush actually happened just a few years before. So I think people had that mentality of, I can find my treasure and I'll be rich. And so everyone started going into rivers and trying to find pearls. Now, to find a pearl, you have to open a mussel. And to open a mussel, you kill it because the actual adductor muscles that hold the shell shut are really strong. And if you try to open the mussel, you will rip those. So to look for pearls, you're killing a lot of mussels, and pearls are very rare, so it's not exactly worth it. The next issue was the pearl button industry. So before plastic was invented, buttons were made out of freshwater mussel shells. So people would collect shells and they would punch these discs to create these buttons. This was a lot worse for freshwater mussels because this is an industry, right? So now we have boats that can access deep water. The scale at which they're collecting mussels is a lot larger. It's unregulated. So they're collecting species that they don't want to use, ones that are too little, too old. So there's a lot of waste happening. And they started noticing that the population started declining. Because of that, they built this station in Iowa called the Fairport Biological Station in 1912. And the idea of that station was to propagate mussels, but first to learn about them, what they are. And the idea was to make the industry sustainable by stocking mussels the way people stock fish. Unfortunately, the building burnt down after 3 years, so that didn't work out, but we did learn a lot, and a lot of our knowledge comes from those initial studies. Eventually, pearl button industry died because of overharvesting, but also because plastic was invented. So now it was cheap to make buttons. So there is, I guess, one good thing about plastic. And then we get into the cultured pearl industry. So the cultured pearl is something that I consider half real and half fake. So these are not plastic pearls that you find in the store, but if you see something called a cultured pearl, it will look beautifully symmetrical and identical to each and every one next to it. And that's because they were purposely created like that. So what you would do is you would collect freshwater mussel shells, you would grind them up, create this little powder, create this little ball called the nucleus. You would then ship them over mostly to China, and they would insert that little ball into a big bivalve, which is effectively introducing that irritant, right? And so they would surgically do that, and they would wait a couple of years while the other bivalve places that little nacre shell around it, and then pluck it out, and ta-da. So you have a real pearl on the outside, but it's not real on the inside. So you're taking something that takes decades and decades to form. You do it in a couple of years. It's fast, it's cheap. And so there you have cultured pearls. Now, this is a legal thing. It's not illegal to collect mussels in many states. But there are some problems. There's a question of whether or not this is truly sustainable because freshwater mussels are so endangered in general. Should we really be doing anything like that? Poaching has been an issue. People never think of poaching happening in Ohio, but Ohio has mussels that are extremely protected. So every state has different laws. In Ohio, you are not even allowed to pick up a mussel without a permit. In other states, that's not the case. So if you're in a state where it's legal and you kind of collected all your mussels and you're looking up at Ohio thinking, ooh, there's this untapped resource, what people would do is come to Ohio illegally to collect mussels. How are they caught? When you're in scuba gear in the middle of the night with bags, it looks a little suspicious. So there have been court cases in Ohio, and they've actually reached the federal level where people cross state borders. So poaching has been an issue. Also, identifying species is really difficult. So even for experts, it can be really hard to identify different species. So you might not always know what you're collecting. Now, with that said, the cultured pearl industry has actually moved away from using freshwater mussels very recently. The technology has developed to the point where they use something else. So this is not really a very current problem anymore, which is a good thing. Okay, so impoundments, the building of dams. This was one of the really, really huge, impactful events, especially as it peaked in the 50s. The building of dams completely changes the river habitat, as you guys probably already know. So you're taking water that was a nice flowing river and you're creating basically a lake, right? You're taking water that was moving, now it's still. The environment was shallow, now it's deep. There's sedimentation problems, right? So when water is moving very fast, it's carrying a lot of silt. When it slows down, all that silt and sand kind of settle. So you have a lot of sedimentation that can suffocate mussels upstream or make it too mucky. Temperature changes. There's also barriers to fish. So if your fish can't get to you, it doesn't matter how healthy you are. If you can't reproduce, that's a problem. Now, the good news about impoundments is that these days we are taking out more dams than we are putting in. So that's moving in a very good direction. Okay. Channelization is another issue. This is where people basically take a nice little meandering, beautiful stream and make it straight, right? So they dredge it, they straighten it. Sometimes it's used for transportation purposes for larger rivers. For smaller streams, it can be used for agriculture. The actual process of channelizing a river kills off mussels. But also you take out all the big boulders and now your sediment's not very stable. So it's harder for the mussels to stay put. That also causes sedimentation downstream. And then if you look at this habitat, there's a lot of microhabitats within that, right? You have the riffles and pools, you have shady areas, shallow, deeper. There's a lot of different habitats for different species. And then you create this one homogeneous habitat that might be all right for one species or two, but you're not going to have the diversity that you had before. Okay, pollution, that's another common one, especially from agriculture. So farmlands put on fertilizer, pesticides, herbicides, all these things, they all wash downstream into the river and get concentrated there. So one way to prevent this is to build these river corridors. So if you have a little strip of forest or heavily vegetated land between the farmland and the river, what happens is all that vegetation slows down whatever is running into the river. So if you have fertilizers or pesticides running down, the movement will be slowed down, but also the plants will uptake a lot of that. And so less gets into the river. And it's actually been shown to really improve water quality a lot for mussels and other animals. Mine runoff can be an issue in certain areas. I find this one interesting because that's a before picture and that's an after picture. So this was a photograph on a website for a consulting company that cleans up rivers. So they're advertising, hey, this river here in Pennsylvania looked like this. So we came in and we restored it. So what happens is when you have an abandoned mine and water runs through it, it takes up all these metals, it becomes very acidic, and it kills off absolutely everything in that river. So all the fish, all the insects, everything is dead. So they come in, they restore the river, maybe they close off the mines, maybe they make a wetland, and it's beautiful and healthy, and the fish swim back and the insects fly back, but the mussels don't just walk back, right? They take a really long time. They're long-lived. They don't really move, and so it take a long time for mussels to get back. So what we sometimes do is we'll propagate mussels that are common, stock them into these areas, sort of to jumpstart all those ecosystem services like water filtration, things like that. Another issue is wastewater treatment plants, right? These are areas that collect sewage and clean the water of our human waste. There is still higher ammonia downstream of those, no matter how hard you try. But we're also finding a lot of contaminants that are not treated. And I'll talk a little bit about that later. But pharmaceuticals, medications, and personal care products, detergents, shampoos, everything that you use gets washed off in the shower or gets peed out, and everything ends up into the river. And wastewater treatment plants are not designed to treat the water for the amazing amount of variety of chemicals, right? Is designed to treat for waste and maybe some ammonia, but we have all these chemicals circulating in water that are not being treated for, that are also affecting wildlife. Zebra mussels. It's Lake Erie, so you have to talk about those, right? So zebra mussels are a very bad invasive species. They are native to the Caspian and Black Sea in Europe. So they initially spread throughout Europe because of channelization. And then they ended up over here in 1985, most likely in ballast water in a cargo ship. So when you have a cargo ship that leaves a port, it will fill a part of its ship with water for balance. And then it will take that water, and as it travels across the ocean and docks somewhere else, it will let go of that water. And that's likely how the zebra mussels got here. Zebra mussels don't bury. They act more like the marine mussels in the sense that they have these little sticky threads, right? So they'll attach to anything hard. They only live for like 3 years. They reproduce like bunnies in the world here. And so they'll attach and massively reproduce and just be everywhere. So that's a cart that's pulled up from a lake, just covered in zebra mussels. We know it causes damage to industries because they clog pipes, which are really hard to clean and replace. For freshwater mussels, they are a problem because if you imagine sort of sandy-ish bottom on a lake with something hard sticking up, like a mussel shell, the zebra mussels don't care that it's a mussel. They just know it's something hard that they can attach to. So when they do that, you can see this part of the freshwater mussel shell is under the sediment. That part was sticking up and it's covered by zebra mussels. The zebra mussels with their sticky threads will basically sew the shell shut and kill the mussel. If not, they'll filter out all the food before it gets to the mussel itself. And so in areas like Lake Erie and the Great Lakes where zebra mussels have become a big problem, they have absolutely devastated freshwater mussel populations. Now, the good thing is that in general, zebra mussels tend to prefer slow moving water like lakes, and most freshwater mussel species tend to prefer rivers and faster flowing water. So there is a separation in that habitat. But like I said, in certain areas like around here, it's been very negatively impacted. Okay, so because of all those reasons, that is why mussels are extremely endangered. So here is a graph showing the status of mussel species in Ohio, but this is actually representative of the United States. It's not just Ohio. But if we take the 60 species that we have and we separate them into categories, the green part here those are considered secure. So maybe a fifth of the species we have. Then we have species of concern. We have those that are threatened. We have those that are endangered. Extirpated means they're not extinct, but they're not found in Ohio anymore. And then we have 5 species that have gone extinct in Ohio. So this is why they are considered one of the most endangered groups of animals, because like I said, this is very representative of the United States. So now that you guys are all thoroughly depressed, let's get into a more optimistic part of the presentation. So now I want to talk about the freshwater mussel conservation facility that I work at. It's called the Waters Aquatic Conservation Center because it was named after my advisor and the original director, Tom Waters. But we focus pretty much solely on freshwater mussels. And we don't specialize in any one thing, although I guess specializing in freshwater mussels is already niche. But we do a lot of host identification. So I mentioned before that mussels can be very specific to what species of fish they can transform on. And we don't know those relationships for a lot of mussels. There's a lot of research that needs to be done there. Somewhat because when you propagate mussels, you need to know what fish to use. But also, if you're doing conservation work outside, if you're relocating mussels or you're trying to stock areas, you need to know that your fish is there because otherwise everything is futile, right? You have to have your fish to be able to succeed. We do relocation, propagation, monitoring. We do a lot of research, right? So we're associated with OSU. We have high school students, undergrad students, grad students work with us. And this is what the middle area looks like, the main room. So we are a flow-through facility, meaning we are next to the Scioto River. We are constantly pumping in water. So water from the river out there goes into these tanks and it gets pumped through our system. We have another one right there. And the reason for this is because mussels are notoriously difficult, if not impossible, to keep in aquariums. The reason is because one, they filter huge amounts of water, so you kind of can't keep up with that, but also because we don't know exactly what they eat. We know they eat bacteria and algae and detritus and all these things, but we don't know exactly. We know that it changes by age, it changes by species. So for us, it is just easier to supply them with a wild source of water. Okay, then we have a host fish identification lab. So we've been operational for about 20 years, and during that time, we have identified 33 fish species for 18 mussel species. So the way we do this is we will find a gravid mussel, meaning that it's a female that's pregnant. We know this because when we open her, and we do this very scientifically, so we don't kill the mussel, and we can see the gills are really inflated, we'll know, okay, so this one has Glaucidia. We can then insert a needle and a syringe that's filled with water, flush out all the Glaucidia into a petri dish. So now we have our Glaucidia soup, and we'll take the fish that we want to try, put them in a 5-gallon bucket, dump the Glaucidia soup in there, swish it around for half an hour. It's very high tech. So we let the mussels attach to the fish during that time. Then we take the fish and we separate them out into these individual little tanks. And in each individual tank, we have water coming in from the top and water exiting out the bottom. So this is the front, but in the back where the water exits, next to each of those little pipes is a cup with a mesh bottom. So whatever goes through gets collected. So after a couple of weeks, if there was a mussel that transformed on that fish, it would not have escaped because it would have been caught in that little mesh. So we get to check the contents of that every day under a microscope. And if we find a mussel that's transformed, we have identified a new host. We also do in vitro propagation. So this is not in vitro fertilization. The mussels have already done that part for us. But this is basically transforming mussels without using a fish. So we do the same thing. We find a gravid mussel, we collect the Glaucidia, but instead of infecting fish with that, we'll put it in a petri dish with the right formula of nutrients, and the little Glaucidia will close by themselves and start transforming. So we think it's in response to the salt. They think they're attaching to a fish, and so they'll close, and they'll take a couple of weeks to transform. Now, there are three major components in culture medium. So one is the basal culture medium. So this is sort of a standard mixture of salts that pretty much anyone that does any sort of culture work buys. We use M199. The second very important part is serum. So serum is blood with the red blood cells removed. Okay. And we use rabbit serum. And that's what provides most of the nutrients, right? All the things that the Glochidia need to transform. And then the third part are the antibiotics and antifungals. Again, everyone uses these because if you have a little dish of nutrients at room temperature, you can imagine anything and everything will want to grow in there. So we want to make sure it's sterile. But it's also a balancing game because those antibiotics and antifungals are toxic to Glochidia. So you want enough to kill the spores, and you're basically trying to slow down the growth of anything else long enough for the mussels to transform without using so much that you're killing off the mussels. So this here is a project that Katie, a grad student, did a few years ago. So she was doing in vitro propagation with a mussel, and she was using that M199 basal medium that I talked about and comparing it to another one, L15. So these are just different concentrations of salts. And she was transforming the freshwater mussel, Lampsilla silicoidea, which is the fat mucket. It's a common mussel. And she wanted to see if there were differences in gene expression in those transforming Glochidia. And so using transcriptomics, which is a method to look at which genes are being expressed, we found really large differences between the mussels that transformed in the different media. And then we also had a couple undergraduate students join us for a research internship in the summer who kind of piggybacked on that idea, and they looked at the transformation success rates between the two and were able to show that mussels transformed higher in rates in one than the other. Okay, so now this is a study that is done by my current grad student, Gabby. So she's actually looking at the effect of the serum. Okay, so that's the blood with that blood cells removed. And I said we use rabbit serum because that's the thing that's commercially available. It's sort of the standard in the field. You could imagine that fish serum would probably be better, right? Because it's closer to what they actually need. And what she's doing, she's transforming two species. So she's transforming the purple wartyback, which only transforms on catfish, and then the fluted shell, which is a generalist and transforms on almost anything. So then she's raising them in serum that's collected from the rabbit, which is our standard, but she's also doing catfish, right? So that's this guy's host, the catfish, and then also carp. Carp is closely related to catfish. And then we go kind of phylogenetically more distant to bass and more distant to sturgeon. And we want to see if the rates of transformation are changing when you go further away from the host, because you can imagine that maybe for the specificist, this matters, and maybe for the generalist, it does not. So she's currently working on that. So I don't have any cool results to show you guys, but I can't show you pictures of us collecting serum because fish serum from largemouth bass is not commercially available. So you have to collect it. So we were lucky enough that ODNR were doing their bass surveys that they do every year. They go out when it gets dark because that's the best time to catch largemouth bass, electrofishing. And so we would collect the fish and we would take blood samples. So you can either go into the heart or into the caudal fin. You might think that this is, okay, blood drawn, no big deal. It's brutal. It is very brutal. I'm not gonna lie. So the fish has to be, not sedated. So you're holding it down, trying to keep it still. It's not easy to poke it right there because it's there's a cartilaginous something, another plate in front of that. And then you have to try to aim for the heart. It is not easy. So she gets to do that quite a bit, which is always fun. And then other work that we do is looking at stress. So when I first started, there was almost nothing known about health for freshwater mussels. So you can't really look at a mussel and see if it's happy or if it's sad, and we just don't know. So I used metabolomics and transcriptomics, so tools that look at the metabolites that are within the blood or the hemolymph muscles, looking at their gene expression, and seeing what happens when we move mussels into captivity or to another creek. So relocation is actually a very common conservation technique for various reasons. Maybe you're building a bridge somewhere and that construction is going to kill off mussels. You need to move them. Or maybe you're propagating them. You need to translocate them somewhere else. It's done very, very often. But it turns out that it is extremely stressful for mussels. So when you move mussels into captivity, which you would think is a very stressful environment and not great, it would be worse than moving them into a beautiful, clean creek. But it turns out that the stress response is exactly the same, and it lasts for about a year. And it's probably because these mussels have grown up their whole lives immersed in one very specific environment, and you move it to another environment, and it is very, very stressful for them. Okay. So I think this might be the last study that I'll talk about. So I was lucky enough to piggyback on to a study that Fish and Wildlife Service did a few years ago, looking at contaminants of emerging concern in the Maumee River. So contaminants of emerging concern is a very fancy term, basically for all the chemicals in our water that shouldn't be there. And they're called that because we're only relatively recently discovering them, and we're discovering them at rates that far outpace the rates at which we can do toxicity tests to figure out if they're bad or what's going on. And they were looking at different areas and putting out cages of fish. And so I joined in and I took mussels from one pond, so one source, and I put them in these cages here in different locations along the Maumee River. And I did the giant floater, and we wanted to see if those different areas, which would supposedly have different mixtures of contaminants, influence their physiology. And I know this is kind of hard to read, so that's just a list showing the most highest concentrations of contaminants in each location. There are things like, I think the number 1 or 2 in every part was an X-ray contrast agent. So I'm guessing there's a hospital upstream. Metformin, DEET, right, in bug spray, caffeine. We found all kinds of things. But just because something is in high concentration doesn't mean that's necessarily the worst for you, right? You can have something in a very low concentration that's much more toxic. So what we did is we compare the concentrations and the mixtures in the specific areas, and then the fancy stats to compare them to the metabolites that changed our mussels. So we can see that certain metabolites are different than in other ones. And so we are able to actually use those stats to rank the contaminants that are the most harmful, or I should say the most impactful, right? That change the physiology of the mussels the most. And so here I show the top 10. And so the number 1 is actually a breakdown product of an insecticide that was banned in 2010. And it's banned because it's very toxic. It's also very persistent, right? It's still in the environment. The next 2 are breakdown products of chlordane, which was banned in 1988. Still around and still the number 2 contaminant that's affecting mussels. Next down on the list, we have an antibiotic. I guess it's very common. People use it. It's in the water. Xanax, an antidepressant, which we know is actually influences their reproductive physiology. It's a neuromodulator, right? So it impacts that kind of signaling. Another insecticide banned in 86, insecticide banned in 88. Then we have bug spray and another insecticide banned in 1974. And then the 10th one was an antibiotic that's used for livestock because there were feeding operations with cattle upstream. So they are fed, antibiotics that then end up into the river. So I think this kind of shows that even when you ban insecticides or things that are toxic, they still linger in the environment and are still very impactful and toxic. But I guess the good thing is if you look at the years, right, they kind of slowly go away after a few decades. It'll take a while. Okay, so that is the end of my talk. So I just wanted to acknowledge the facility, is a collaboration, right? I work for Ohio State University, but the facility is a result of a collaboration between the Columbus Zoo and Aquarium and ODNR, so the Division of Wildlife. So those are our big partners. We also work Fish and Wildlife Service, and we just have a lot of students to help us and a lot of other people. So thank you. Do you guys have any questions? Yes.
So you're just talking about like all the like behaviors for the different mussels that have one of those like um words like that behavior. Has there been any research done like about like you said about the crayfish or anything like that? How they know like how crayfish moves and everything like that?
So that gets into evolution, right? So they don't know. They don't know what a crayfish looks like. They don't even have eyes, so they can't really see. But imagine if, you have a certain characteristic that attracts fish to come to you. Maybe like it looks like something is kind of edible and the fish might eat that. Then those babies will then grow up and survive and have the genes for that particular trait. And in the next generation, if you have a little bit more of those traits that interest fish, more of those will survive. And so just through that process, through an evolutionary time period, you develop something that looks like a crayfish without the mussel knowing it is, right? It just knows that or it works, right? Or it doesn't even know that really, it just does it because that's what's instinctual to it. Yes.
Generally, how old are the species and how are they beneficial to their freshwater environment?
So how old you mean like what their lifespan is?
So, like how long they've been in their current form?
Oh, okay. So they've been around since the dinosaurs. The freshwater mussels themselves, they have an ancestor that's marine. All bivalves that are in freshwater had a marine ancestor. There have been the separate invasions into land over time, but we know that these have been around since the Triassic period. So that's why they're a very unique group of animals. And then the second thing you asked me is how they help the ecosystem.
Yeah.
Okay. So they are huge filter feeders, right? So that means they clean our waters. I like to think of them as livers because your liver cleans your blood and cleans your body. But if you abuse your liver, it's not going to be happy. And we're abusing our rivers, right? So I can think of it that way. But also because it cycles the nutrients in the water column, it has a really big impact on nutrient cycling. So when it uptakes nutrients, it can then excrete them next to it on the bottom. That creates more food for algae, which then creates more food for other insects. And you can actually find that the diversity of insects in mussel beds is higher than where there aren't any. So it does influence like the ecosystem processes and the food chain and so on. Mhm. Yes.
From the rivers and stuff like that.
I am not a toxicologist. I have no idea. I don't know. I think that maybe there isn't, and you just have to wait for them to break down further. I know that they're persistent in that sense. Maybe someone else knows. Or someone's raising a hand. Oh, you just had a different question. Yes, I don't know. I don't know. So I think that I'm sure there are chemists out there that are already working on it. I bet if I went online and searched for research studies on that, people probably are already looking into that. And I think that we know that these chemicals are bad. I don't think it's a secret, maybe necessarily which ones are impacting mussels more are. But I'm sure that you could, yeah, I'm sure people could try, but I feel like people probably already doing some kind of work on that. Mhm. Yes.
In your research, have you come across any evidence that mussels cohabitate with Ingoldian fungi or hypomycetes to help with this filter feeding ecosystem?
I don't know. I really don't know.
It's the second
Yeah.
question.
Mhm.
Since this is the most endangered, well, family or genus of species in North America. Is this being utilized with the EPA to help reverse the combined sewer overflow system problem that the Ohio River experiences?
So not in Ohio, but Chesapeake Bay does that. They actually have a propagation center specifically for mussels to clean the water.
Okay.
But that's actually an exception because freshwater mussels are so endangered that you kind of want to restore the populations themselves before using them to clean the water, right? So yes, you can. I don't think anyone is doing that in Ohio. But places like Chesapeake Bay, I believe, have built a facility just for that purpose, and they got funding, I believe, from the government for that purpose because they don't care about the endangered species; they're raising the common ones. So it's actually a different focus, right? So they're propagating all the common species because they want to use them for filter feeding. But we focus more, we have done some common species because when, for example, there's a spill in a river, let's say some company does it accidentally or maybe on purpose and they're taken to court, you can imagine they have to pay these huge fines. Some of that money trickles down to us so that we can then restock common mussels. But you wouldn't do that with endangered animals, right? Because you wouldn't want to raise threatened species and then put in to clean the water for you because they just wouldn't really work out that way. So it's kind of two separate areas, but I don't believe anyone in Ohio is doing specifically that.
I guess I was curious as using the incentive that they are endangered and then using that legality to encourage legally cities to fix their sewage problems.
Well, I don't know about that specifically, but that is a reason. I mean, people know that. Yeah, you raise an interesting point, right? Because I don't think anyone has specifically used mussels as a reason to. And I'm not really involved in lobbying or things like that. So I'm not exactly sure, but I think a lot of like nonprofits, they actually do work with rivers and have visited our facility and it's funny because there is someone that works with freshwater mussels, but is in the Nature Conservancy that is trying to fight the spread of this town growing near us that is going to also need a larger sewage treatment plant. And they were trying to use mussels in that defense. And so they was talking to me and trying to get ideas. I'm not really sure how successful that was in that sense.
I know there's some politicians that have tried to appeal protections for mussels, like Ken Paxton, Texas, and I think someone in Indiana as well in the past. So I was just curious if that was or that was access to fix our problems versus just endangering species.
Right. As far as I know, not specifically. I don't know specific examples like that. I think that in general, it's hard to get people to care about mussels. I think hopefully after the last 40 minutes, I have made you guys care, but you can see like it could take a little effort, right? And so I think if people don't really understand how important they are or even have a connection with them, right? Then it's difficult to use that. So we focus a lot on public outreach and education, especially since we're associated with a zoo because as cheesy as it sounds, but like teaching people about them and making them sort of understand and connect is important because eventually, if you're going to use that reason, the people have to care about mussels, right? And yeah, usually they care about what mussels do for you instead of what you should do for them. So, yeah. Yes, I think so. In the back, actually had a question for a while. Yeah. So I'm sure other people in this room in the back are better at answering this than I am, but I would say from what I know, and you guys can correct me, no. I think I think the best that you can do is try to prevent the spread. But I'm not sure I think people have tried things, but I don't think there is. Is there?
Equinox.
Okay.
They can control mussels in very small
areas. So like something like Lake Erie or the other Great Lakes is much bigger, right? But there is a group, I think it's within the Great Lakes Commission called the Invasive Mussel Collaborative that have done studies on spawning reefs for lake trout, where they have, I don't know if it's a curtain type thing, but they've isolated spawning reefs and put, I think that chemical or another chemical that actually kills zebra mussels off for long enough, and they're trying to see if that will clean off the reefs enough to allow the fish to spawn. Obviously, they're going to recolonize that, but we'll see if there's a functional time that they can.
And then my question is, does that impact freshwater mussels?
I don't know that there's any left.
Like, good point. Yeah. Right. Yeah. So I think my biggest concern is actually the golden mussel now. So this is a mussel that is ecologically very similar to zebra mussels, and it is invasive in Brazil. And they just discovered it in California, I think a couple years ago, which is very bad news because from what I know of the mussel, it's similar to a zebra in every way, except it also likes flowing water. And that makes me nervous. So I don't know. I don't know what they're doing with that over there, but I wonder if that's going to be our next zebra mussel. We'll see.
Take one more question, which is from online. What are the natural predators of mussels?
Muskrats are probably the biggest one. So if you walk along the river and you find a little pile of shells, unless it was made by a human, it most likely is a muskrat midden. So they will kind of take the shells and pry them open. Carp eat mussels. Sometimes it's a good thing because that's how the mussel is attached to larvae. But I know that they can also eat some freshwater mussels, but there's not a lot. There's not a lot of predators. Yeah. Okay. Well, thank you so much.
I want to thank everyone who joined online. And if you want, you can join us next week. Next week is the NOAA and Ohio Sea Grant harmful algal bloom forecast for Lake Erie. And the speaker next week will be Dr. Nate Manning from Heidelberg University talking more about nutrient loading into Lake Erie. Thank you all for joining and hope to see you next week. Bye.